Today we will discuss about heme biosynthesis. Here we will know what is heme, what is porphyrins, then heme biosynthesis steps, then regulation of heme biosynthesis. Now we have heard of the substance hemoglobin. Where do you find hemoglobin? Where do you find? In RBCs, no? You find hemoglobin. Now in hemoglobin there are two parts. One is heme part and another is globin part. Heme part is the iron containing prosthetic part. Rather should I say ferroprotoporphyrin. Heme is the ferroprotoporphyrin. That's why we are going to see what is porphyrin. Ferroprotoporphyrin, heme. It is the prosthetic group and the globular protein globin. Okay. Now hemoglobin is composed of heme and globin. Heme is red color. Heme is red color. That color is provided by the protoporphyrin. Provided by the protoporphyrin. How it is provided, we will see later. Now heme has 4 pyral rings in cyclic form. These pyral rings are together called protoporphyrin. Protoporphyrin 9. It is also called protoporphyrin 3. Same thing. Previously the nomenclature protoporphyrin 9 was used. Nowadays 9 also used, 3 also used. Whatever it is used, it is for the protoporphyrin, it is same. Protoporphyrin 9 and protoporphyrin 3. That protoporphyrin, in its center, it contains iron in ferrous form. These are some molecules which contain heme as prosthetic group. We will see it later in detail. Now, before we proceed for heme synthesis, let's see what is porphyrinogen and what is porphyrin. Porphyrins, they are cyclic compound. This is the pyral ring. Okay. Now in so this is a cyclized pyral ring. Here it is in cyclized form. Then fusion of pyral rings are there linked by methynyl group. Okay, they are deeply colored, and the porphyrin which is present in him is called protoporphyrin 9. Now what is porphyrinogen? Porphyrinogens are the precursor molecule for porphyrins. Porphyrinogens are the precursor molecule for porphyrins. Here is protoporphyrin 9. This is porphyrinogen. Okay. Now porphyrinogen also having cyclic pyrrole rings. 4 numbers of pyrrole rings are there. However, they are linked by methyl group. You see, there is no double bond. There is no double bond, but here it is, double bond is there, methynyl group. Okay. So this double bond, this conjugate double bond in pyrrole rings, conjugate double bond in pyrrole rings and linking methynyl group is responsible for producing color in the porphyrins. However, there is no double bond, conjugated double bond in the porphyrinogen, so they doesn't possess any color. Now the question comes for your exam: explain why porphyrinogen are not colorless, however their derivative porphyrins are having deep color. Okay, their derivatives have color, but they are not having color. Explain why. This is the question. Now, structure of porphyrins, we have already told 4 pyrrole rings. This is 1, first, second, third, fourth, connected by methynyl bridge. Methynyl bridges are denoted as alpha, beta, gamma, delta, and it has 8 substitute hydrogen atom which can be substituted. They are in position 1, 2, 3, 4, 5, 6, 7, 8. Now, if the substituent group in these areas are symmetrical, they are called type 1 porphyrins. Type 1 porphyrins symmetrical substituent group, and type 3 porphyrins, there is asymmetrical substituent group. Okay. Now, see, while dealing with the structure, it is very clumsy type of structure, no? Very difficult to write these things. So, father of porphyrin chemistry, he gave a model of porphyrin structure. This is in this model, he proposed this model, and in this model the porphyrin ring is replaced by a bracket. It is replaced by a bracket. Now here you have seen the 4 pyrrole rings. The first pyrrole ring is represented by a bracket. Second is again represented by a bracket. Third is represented by another bracket. So this is the Fischer model of porphyrin structure. Okay. This is the first. This is second. This is third. And this is fourth. These are the substitution areas: 4, 5, 6, 7, 8. So, what will happen in case of type 3 porphyrins? The substituent group between 8 and 7 will be asymmetrical. Okay. Now, if it is A and P, A, P, A, P, this is for understanding purpose. so that you can understand what is the type 3 and type 1. A, P. So here this is symmetrical, so it is type 1. Now in case of if we write like this, AP, again here P will come first, A will come later. P first, AP, PA. So it is symmetrical, that's why it will be type 3. There is no 2. So now what are porphyrins? They are cyclic compound having 4 pyrrole rings held together by methynyl bridge, metal ions. Porphyrins you may get short note, or you may get as question also short answer, short answer type question, what is porphyrin? So you will mention these points. Cyclic compound having 4 pyrrole rings held together by methynyl bridge, metal ions can bind nitrogen atom of pyrrole ring to form a complex. Here it can bind here, okay, between, it will replace H and H, this nitrogen atom and this atom will be bound to this one, ferrous ion. Naturally occurring porphyrin contains substituent group replacing 8 hydrogen atoms. If it is symmetrical, it is type 1, it is asymmetrical, it is type 3. Presence of conjugate double bond in pyrrole rings, conjugate double bonds in pyrrole rings, here, they gives responsible for color, okay. Now physiologically it is the type 3 porphyrins which are important. Type 1 porphyrins, they are only serve as precursor, okay. Now in case of porphyrins, they have a special character called Soret band. Soret band is nothing but when they are exposed to a light wavelength of 400 nanometer, they absorb the light and emit orange-red fluorescence. Why it is helpful for us, Soret band? Because in case of porphyria, due to the deficiency, due to the problem in synthesis of heme, there is different type of porphyria, and those porphyrinogens, they are excreted in the urine. So we subject that urine or blood to this wavelength of light, and we can identify the porphyrins excreted in the urine. Now this again is proportional, the absorbance of light is proportional to the concentration of porphyrins, okay. Now we can quantify as well as we can identify the porphyrins. Now some of the porphyrins, uroporphyrins, just see uroporphyrins. This is uroporphyrin 1. I told it is type 1, type 3, what is the difference? So in type 1 uroporphyrins, it is AP, okay. This is acetate and propionate. They are bond in the substituent groups as substituent group in the 8 places as symmetrical form. So they are type 1 porphyrins. However, here you see P comes before the A. So this is uroporphyrin 3. Here the thing is asymmetrically substituted. Coproporphyrin, same thing. Only thing is that here in place of acetate, methyl group is added. Okay, others are same as that of uroporphyrin. Now in heme, it is the protoporphyrin which is present as porphyrin. Here the methyl group is as it is there, but in the first position and second position and fourth position, the propionate is replaced by vinyl group. Vinyl group. But in case of 6th position and in 7th position, as because it is type 3 porphyrin, the substituent group in the 7th position will be propionate. Difference is here all 4 groups in 2nd, then 4th, 6th and 8 or 7 are not same. Okay, in 2, 2nd and 4th it is vinyl, and in 6th and supposed to be, if you take it as 8, it is propionate. But as it is group type 3 porphyrin, so in place of 8, the propionate group is attached to the 7th position. Okay, it is asymmetrically, it will be asymmetrical in position. Okay, so it is a type 3 porphyrin. Only type 3 porphyrins which contain asymmetric situations are physiologically important in human. So along with the in this protoporphyrin, the iron is added in the center position to give ferroprotoporphyrin or heme. Now what is heme? Heme is nothing but it is a derivative of porphyrins. It is cyclic. Porphyrins having cyclic compound formed by fusion of 4 pyrrole rings linked by methynyl bridge. Don't confuse methynyl. Okay, methyl and methynyl. Methynyl bridge. There are 4 methyl, 4 methyl groups, 2 vinyl and 2 propionate side chain substituent. Okay, side chain group. Iron is in center of protoporphyrin, so heme can be told as ferroprotoporphyrin. Now, what is the function of heme? As it is the prosthetic group of hemoprotein, the function of hemoprotein is the function of heme. So, some of the hemoproteins, they reversibly bind to the oxygen like hemoglobin, myoglobin, neuroglobin, cytoglobin. These are nothing but the oxygen is bound to the heme component. So, hemoglobin transfer of oxygen and carbon dioxide, buffering action, then myoglobin, oxygen reserve in muscle, neuroglobin, oxygen reserve in nerve, then cytoglobin, oxygen reserve in multiple tissue. These are the functions. If you are asked what are the functions of heme, write these things as function. Now some of the enzyme catalase, this converts hydrogen peroxide into water. In metabolic reaction, many reactions there are hydrogen peroxides are produced. These are injurious to the cell. That's why it is immediately catalyzed into converted into water by catalase. Peroxidase, then cytochrome P450, monooxygenase. These are detoxification. They detoxify the toxins in the body. Then these are responsible for steroid hormone or steroid metabolism. Then nitric oxide synthase, generation of nitric oxide, cyclooxygenase in prostaglandin synthesis. Next comes your complexes and mobile carriers of electron transport chain. Cytochrome C oxidoreductase, cytochrome C oxidase, cytochrome C, like that. So many heme containing enzymes are there or carrier proteins are there. So these are the functions of heme as hemoprotein. Now, where does heme synthesis occurs? Site. These are the erythroid precursor cells of the bone marrow where heme synthesis occurs. About 80%, 85% of the heme synthesis occur in erythroid precursor cells of the bone marrow. And hepatocyte, it can synthesize about 15% of heme in hepatocyte. Now, which part of the subcellular organelle it is synthesized? Mitochondria and cytosol. These both takes part in heme synthesis. We can divide steps of heme synthesis into 8 important reactions. Out of that, first, 6, 7th and 8th reaction, they take place in mitochondria. And reaction 2nd, 3rd, 4th and 5th reaction, they take place in cytosol. Remember, matured RBC cannot synthesize heme. Why? Anyone please, why? Matured RBC, it is important for production of RBC, but matured RBC cannot synthesize heme. Only it contains heme as hemoglobin. Batao, please tell why. You should be able now. Yes, because of the absence of mitochondria. So that's why it cannot synthesize. Very important. Explain why mature RBC cannot synthesize heme, though it contains heme as hemoglobin. Heme synthesis proper. Now, very difficult to remember, so we will try to remember some mnemonics. You might have seen some great doctors palpate heart under cover to produce heme. So this is mnemonics. We'll try to remember. Now, in first step, succinyl CoA is condensed with glycine amino acid by delta libulinic acid synthase. This is the committed step. the first step is the committed and rate limiting steps. This ALA synthase that requires PLP as cofactor, and carbon dioxide is released, delta amino linoleic acid is synthesized. This occurs inside the mitochondria, first step. Now this ALA, it is carried to the cytosol. In second step, two molecules of delta amino linoleic acid undergoes condensation with removal of water. Enzyme is LR dehydratase. It contains zinc containing enzyme. It is also called porphyrinogen synthase. Porphyrinogen synthase. This enzyme, this can be inhibited by lead, and this lead, it also can inhibit the last step of the heme synthetic pathway, that is ferrochelatase. So both side it can inhibit, producing anemia. Now in the third step, 4 molecules of porphyrinogen, they combine to form hydroxybilane. Hydroxybilane is a tetrapyrrole. Porphyrinogen is a monopyrrole compound having single pyrrole ring, and hydroxybilane is tetrapyrrole compound. We know porphyrins are cyclic, having cyclic pyrrole rings. So we'll see how it is formed into cyclic pyrrole rings. Now this porphyrinogen, 4 molecule of porphyrinogen, they are converted into hydroxybilane. 4 molecule of ammonia is released. The enzyme responsible for this is porphyrinogen deaminase. Commonly it is also called uroporphyrinogen 1 synthase. Uroporphyrinogen 1 synthase, or it can be called as hydroxymethylbilane synthase. So same enzyme is known as 3 names. This is the only one enzyme. They can be told in 3 names. So whatever you tell, it will be correct out of these 3. Defect of this enzyme causes acute intermittent porphyria. Porphyria you may get as long question or a short note. Porphyria. So different types of porphyrias are there. So it is one of the porphyria, acute intermittent porphyria. This is the most common acute form. This is most common acute form. Others may not be acute form, but they are commoner. Okay. So this is common acute form of porphyria seen in the clinics. Now this hydroxybilane next converted into uroporphyrinogen. See, very easy to confuse, no? This is porphyrinogen. After that, whatever comes, it is porphyrinogen, not bilinogen. So it is uroporphyrinogen. This is porphyrinogen. It is uroporphyrinogen, not urobilinogen, not urobilinogen. This is uroporphyrinogen 3. Sorry. So it is the enzyme responsible for this is uroporphyrinogen 3 cosynthase. Uroporphyrinogen 3 cosynthase. Okay. Now what happens here? Uroporphyrinogen this hydroxybilane, which is linear tetrapyrrole, is converted into cyclic form of tetrapyrrole ring here. Here, the first, second, third, fourth, and substituent groups are acetyl group and propionate group. Acetyl propionate, as because this is type 3 of porphyrinogen, the here asymmetric is there. The P is supposed to be here, A is supposed to be here, but because of this type 3 porphyrinogen, the 7th position comes propionate and 8th position comes acetyl. Why it is? Because it is only the type 3 which is physiologically important. It can take part in synthesis of heme. That's why it is necessary. So now uroporphyrinogen 3 is converted into corpoporphyrinogen 3. What happens here? This acetyl group is converted into methyl group. So four methyl groups will be acetyl group will be converted into methyl group. That's why 4 carbon dioxide is released. So this enzyme is uroporphyrinogen decarboxylase. Now in uroporphyrinogen 3 cosynthase deficiency, the porphyria develops is congenital erythropoietic porphyria. Congenital erythropoietic porphyria. In case of uroporphyrinogen, porphyria cutanea tarda. Porphyria cutanea tarda. Okay. Now, when this enzyme is blocked, what happens? This hydroxymethyl is spontaneously converted into uroporphyrinogen 1. Uroporphyrinogen 1, then with the exposure to light, it gets converted into uroporphyrin. It can spontaneously be converted into corpoporphyrinogen 1 by uroporphyrinogen decarboxylase. Then on the exposure of light, it gets converted into corpoporphyrin. So both these will be excreted in the urine, and they can be measured to identify the type of porphyria. Now, the same thing what I told you. Now, heme synthesis, this corpoporphyrinogen, up to this corpoporphyrinogen, it was reaction occurred in cytosol. Now, the step 6, 7, 8, it will take place in mitochondria. Remember, these oxidases, okay? These oxidases, enzyme oxidases and ferrosilase, they are present in mitochondria. So, they have to, this corpoporphyrinogens, they have to transfer, they have to go inside the mitochondria. So, rest of the reaction take place in inside the mitochondria. So, in corpoporphyrinogen 3, they will be converted into protoporphyrinogen 9 with the help of corpoporphyrinogen oxidase, coproporphyrinogen oxidase, coproporphyrinogen oxidase. So, here what happens? The propionate group, 2 propionate group of position 2 and 4 will be converted into vinyl group. Okay. Now it is protoporphyrinogen 9, also called protoporphyrinogen 3. Same thing, protoporphyrinogen 9 or protoporphyrinogen 3, because here you see in position 7 propionate is there. Then protoporphyrinogen 2 This will be converted into protoporphyrin with the help of enzyme protoporphyrinogen oxidase. Okay. So this will introduce this double bond in the ring structure and make it possible to produce color. Okay. After that, it is the ferrochelatase which will incorporate iron as ferrous form in the protoporphyrin ring and produce heme. So, in case of lead poisoning, anemia is produced due to the inhibition of the ferrochelatase. Now, in case of porphyrinogen oxidase deficiency or dysfunction, the porphyria which is produced is hereditary corpoporphyrinogen porphyria. Then, if protoporphyrinogen oxidase is deficient, then it will produce variegate porphyria. So, almost in all the enzymatic reactions, some or other porphyria is there. So, you have to remember it. So, that is why remembering the enzymes in the pathway, we have just simplified here. Sorry. First is ELA synthase, second is from ELA to porphyrinogen ELA dehydratase, then porphyrinogen to hydroxymethylbilane. This is uroporphyrinogen 1 synthase. Then from hydroxymethylbilane to uroporphyrinogen 3, it is uroporphyrinogen 3 co-synthase. Okay. Now from uroporphyrinogen 3 to coproporphyrinogen, it is the uroporphyrinogen carboxylase. Okay. Now from coproporphyrinogen to protoporphyrinogen, it is coproporphyrinogen oxidase. Now from here, from the protoporphyrin to porphyrin, it is protoporphyrinogen oxidase. Remember, oxidases and ferrochelatase, they are present only in mitochondria. So, okay. Now ferrochelatase will add iron to this, heme will be synthesized. Okay. Now just in short, remember these two. Remember these two mnemonics, you will be able to remember the pathway. Now this is in short the succinyl mitochondria. What is that? Heme synthesis. Succinyl CoA and glycine in mitochondria. Delta aminolevulinic acid is synthesized. Then ELA is carried to the cytosol with the action of ELA dehydratase. Porphyrinogen is synthesized. Then 4 molecules of porphyrinogen they form a linear tetrapyrrole compound hydroxymethylbilane with the help of enzyme porphyrinogen or uroporphyrinogen 1 synthase. Then uroporphyrinogen 3 is produced from hydroxymethylbilane with the help of enzyme uroporphyrinogen 3 co-synthase. Then coproporphyrinogen 3 is produced by the enzyme uroporphyrinogen decarboxylase. After that, this coproporphyrinogen is transported again inside the mitochondria and they are acted upon by the coproporphyrinogen oxidase to produce protoporphyrinogen 9 and which is converted into protoporphyrin by protoporphyrinogen oxidase. Ferrochelatase, it incorporates iron in the protoporphyrin to produce heme. Now see, This is the committed and rate limiting step of heme synthesis. This delta amino libulinic acid synthase, it has two isoenzymes. One is LAS1, LAS2. LAS1 is present in all tissues and LAS2 in erythropoid specific cell, means in the bone marrow. This LAS1, this is inhibited and regulated by many factors. However, LAS2 is not interfered, its action is not interfered by any other external factors. Okay. Now, PLP is required for the this enzyme, delta amino libulinic acid synthase. Now, deficiency of PLP produces anemia, but not porphyria. That is sideroblastic type of anemia. Regulation of LA synthase 1 in liver. That means other than erythropoid precursor cells. So, three mode of regulations are there: repression, allosteric inhibition, and compartmentalization. Please note it. ये बहुत important है, very easy also, but if you don't concentrate, then so in case of repression, what is repression? It is the expressing out the gene, repression. Okay. Now, heme itself act as co-repressor. Co-repressor, it binds with aporepressor and make holorepressor. Holorepressor is the main substance which can regulate the gene expression. So, heme, it binds with aporepressor and forms holorepressor. This holorepressor binds with DNA, thereby decrease in transcription of mRNA for LA synthase. When heme as holorepressor binds with DNA, it inhibits. Okay, it inhibits. But when heme is deficient, then there is no holorepressor, so there is no repression. Rather, there is de-repression. Okay, de-repression is there. So, in de-repression, there will be increased heme synthesis. In repression, there will be less heme synthesis. So, in transcription of mRNA synthase, in case of allosteric inhibition, heme, the ferrous ion of heme is oxidized into ferric ion. So, heme is converted into hematine that inhibits LA synthase. So, there is decreased heme synthesis. Now, compartmentalization. A part of the reaction occurs in the mitochondria, a part of the reaction occurs in the cytosol. So, here heme converted into hematine will inhibit LA synthase from cytosol to mitochondria. So, this enzyme is inhibited from translocating to mitochondria, thus inhibiting the synthesis of heme. Okay? There are some substances which stimulates LA synthase activity. These are steroid hormone metabolites, ethanol, barbiturate. They increase activity of LA synthase, thereby increasing the heme synthesis. Now, regulation of LA synthase in bone marrow. In erythroid precursor cell, it is the LA synthase isoenzyme 2 is functioning there. So it is not induced by drugs that affects synthase 1. So it is not induced by drugs, it is not inhibited by any other factors. So no feedback inhibition of heme is heme formation in erythroid precursor cells. So from here you have to know what are porphyrins. Porphyrins, as short note, give the few words of porphyrin, what I have told you here. Porphyrins, try to mention these points, and here if you can give the Fisher model of diagram, not this one. If you can give this one, it's well and good. If not, then try to give. If you don't give, then also you will get 90% marks. Okay. Then you will get explain why. 2 questions: why porphyrinogens are not colored substance. However, porphyrins are colored substance. Then explain why mature RBC cannot synthesize heme, though it contains heme as hemoglobin. Solid band as such it will not come, but you have to know why it is important. Function of heme, if it comes, your competency it is mentioned. Function of heme you have to know. So function of heme means indirectly it is function of hemoproteins. Okay. So we have mentioned here. Now regarding different types of porphyria, we will have a second class. There we will discuss in detail, because porphyrias are very important. Here also you will get one or other porphyria as short note. So thank you.